Welcome back, everyone, to our afternoon session. I can't wait to see how many more exciting interactions we'll have. In the previous session, we talked about how important it is to keep the patient in view when we think about discovery. And many researchers and clinicians in this community have, of course, been working across the MIT hospital boundary for a very long time. But what has often been missing is the path, the connective tissue, as it were, that allows these collaborations to emerge and grow more systematically, where engineers, scientists, clinicians, and ultimately patients can shape the problem together from the beginning. This is one of the things that HEALS is trying to build. And this next session highlights 4 initiatives that are helping make that possible. First, the Hood Pediatric, first, the Hood Pediatric Innovation Hub, which is working to ensure that children who are often underserved in medical innovation benefit from the same level of technological creativity that we apply to adult medicine. This initiative is led by Professor Laser Edelman and Joseph Vrasica. The next initiative that will be presented is the MIT Program for Health Sciences and Semiconductors, which explores how advances in microelectronics can enable entire new approaches to diagnostics, monitoring, and treatment. This initiative is led by Thomas Palacios. Third, we'll hear from the MIT Mass General Brigham Seed Program, which is building new bridges between MIT researchers and clinicians across the MGB hospitals to catalyze collaborative and innovative projects. This is led by Alex Shalik and Emery Brown. And fourth, menstruation science, which lays the scientific foundation for sex-based precision medicine, which is an area of health research that has historically received less attention than it deserves. Each of these initiatives is different, but they share a common goal, namely creating pathways where discovery and clinical reality shape each other much earlier in the process. To begin, it is my pleasure to welcome Joe Vrasica and Laser Edelman, who will introduce the Hood Pediatric Innovation Hub. And at the end of the session, we're fortunate to have Professor Paula Hammond, Institute Professor, Dean of the School of Engineering, who will moderate the discussion. Thank you very much. Joe and Laser, the stage.
Hello, and thanks for having us. Elazar and I today would like to talk about how we might accelerate pediatric innovation. The Hood Pediatric Innovation Hub was formed to try to help level the playing field for pediatric innovations. Pediatric innovators work in an environment where markets are small and commercial interest is somewhat limited. However, the opportunity there to improve healthcare is quite immense. Let's start by giving you some numbers. One of the reasons we are here is that about 1/3 of mothers need to travel more than 90 miles to get to a maternal unit. Infants born without maternal care have a 14% increase in the risk of death. That's a big number, and one that we think we can address. 20% of approved healthcare algorithms include data from in their pediatric AI models. So think about this. AI is changing the face of healthcare, and models that help us decide what to do for patients, only 14% of approved models include pediatric data. It's a huge problem that we have, that pediatric data is scarce. We hope to overcome that. 10% of the population is over 65 years of age, and major innovations for that population, which I really think should be treated very carefully and very much with care. But when you treat a 65-year-old, you get about 13 years on average of improvement of life. If you treat a child under 18, you can get up to 78 years of impact on the quality of life and on impact on society. There's a big difference, and I think thinking about this carefully, we should address pediatric innovation more routinely in our innovation world. So to get started, the Hood Foundation gave a generous donation to MIT to start the Hood Pediatric Innovation Hub here. Hood was started in 1846, and the initial founders of Hood, which was a milk company, Charles Hood and Harvey Hood, were microbiologists. They noted that children were becoming infected with tuberculosis from their product. And they started some of the first work in pasteurization of milk. And as you can see here, this is Boston Children's. They had their own herd of cows free of tuberculosis in order to give milk to children at the hospital. And it was just on the other side of Longwood Avenue. I think there's a parking garage there now. But at any rate, no need for cows anymore in the field because Hood started the process of pasteurizing milk. In 1942, the Hood Foundation started to establish a network of pediatric innovators. And they've been funding pediatric innovation since then. So over 800 funded pediatric innovators have been set out into the wild by the Hood Foundation. This network forms the basis for our networking in the Hood Hub as well. So we benefit from those 80+ years of innovation leaders that have been set out to become leaders around the world. And they're part of our network already made here at MIT. So this is just a graphic that says, with world-class innovation ecosystem that we have here, with high impact education resources, and with an entrepreneurial milieu, we combine that with the Hood Foundation to create the Hood Hub. The Hood Hub will be bridging the gap between ideas and the bedside. It's a kind of unique approach to innovation here at MIT because this resource that we're building is outside in rather than inside out. We are considered to be neutral ground when it comes to pediatric innovation. There are a number of pediatric innovation centers around the country. Many of them are associated with children's hospitals, and they really work on their own portfolio to extend the portfolio of that children's hospital into the community. Our goal here is to be neutral, to bring innovations from the outside in, to bring innovations, of course, from MIT to the table, and to have a global reach. We have extensive institutional experience in innovation and creative support, of course, and a broad technical and medical expertise in the hub and around MIT. And HEALS is the example of how this happens, right? So people in HEALS all have some focus on medical research and development and innovation. This is what the program looks like. It's centered around 4 pillars: mentoring and education, innovation infrastructure, so creating the infrastructure to take a great idea and bring it right through to the end, to the marketplace. Data and AI, I mentioned the dearth of pediatric data, and building an innovation community with our ready-made Hood network. Here's what the programs look like as of today. An education program. We've started with 19 Catalyst Fellows this year, who many are focused on pediatric innovations in the MIT Catalyst program. We're building innovation workshops. We had one in last July. We hope to have another innovation workshop this July. And building other innovation programs around pediatric innovation. We're building an accelerator, which we'll talk about the next time we meet, and funding support, which we'll talk about a little bit more in a couple of slides. Building mentored support for translation of ideas to the bedside. Prioritizing efforts that will leave enduring artifacts for future innovators in pediatric healthcare. This is how funding happens within the Pediatric Innovation Hub. There are will be catalyst ideas There will be mentored ideas and then direct proposals. Today, we're going to talk about direct proposals. So you're the audience will be the first to hear that for 2026, we're announcing a limited number of innovation awards that will range from $75,000 to $150,000, inclusive of overhead. The typical award will be about $100,000, project duration about 12 months, and follow-on support perhaps for very promising continuation projects. Our areas of focus, and these are not all-inclusive, these are our current areas of focus: artificial intelligence and machine learning and computational pediatrics, sensing and wearables and data sciences, devices and diagnostics, and health policy, ethics, regulation, and building networks in the community to help reach children that don't have easy access to care. Who can apply? Everybody. We have no geographic or organizational limitations. Teams can come from academia, from clinical institutions, from industry, and the application process will be simple. A white paper, 3 pages for the initial stage 1 application, and then selected teams will be invited to develop their white paper into a full proposal. So this is our first announcement for this, so spread the word. This will be going on for another month and a half before we close the applications. And my expectation is that this will bring to life more of our intent to provide enabling resources and collaboration across MIT and across multiple institutions outside of MIT to bring great ideas to us so that we can help them across the bridge to the bedside. Thank you.
It is absolutely great to have you all here and to be able to share with all of you the work that we have been doing under the new MIT program for health sciences and semiconductors. My name is Tomás Palacios. I'm a professor in electrical engineering and computer science here at MIT. And with my colleagues, Professor Ahmad Bahai and Professor Alex Shalik, I have the honor of co-leading this new initiative. I think the key message that I believe we all share is that the future of healthcare will rely on finding ways to make healthcare more affordable and more personalized. And the question is, how do we accelerate that transition towards a more affordable and personalized healthcare? And we believe that part of the answer is going to be by bringing in more ideas, concepts, and technologies from microelectronics and semiconductors and the chip industry, which has been able to reduce the cost of a computer by millions of times over the course of a few decades. And the good news is that this is starting to happen. There is a revolution happening in personalized healthcare, as we can see from the different products that are starting to come to the market, all the way from new genome sequencing techniques to glucose monitoring patches, or the use of microfluidic chips to accelerate drug discovery. And this is just accelerating. And it is accelerating because of the unique convergence of 3 technologies that is happening right now. And those 3 technologies are the development of new materials that are absolutely critical for the invention of a new generation of sensors and biosensors to be able to measure physiologically relevant signals in our body. So new materials. The second big trend is what is called heterogeneous integration of those materials with semiconductor chips. I come from the semiconductor world. And then the third trend that is happening is, of course, AI, which is able to extract the information out of all the new data that the sensors are generating. So this convergence of new materials to enable sensors with heterogeneous integration to be able to use the scaling of microelectronics plus AI was not here 5 years ago, not even 3 years ago. It's happening right now. That's why the timing is just perfect. And we believe that MIT, in close collaboration with all our partners, it's uniquely positioned to drive this new revolution in more affordable, personalized healthcare. Because our community, all of you, are really at the front edge of what is needed to make this happen. We have some of the leading groups on semiconductor and edge AI in the world. We have amazing clinical and translational expertise and facilities. Through collaborations with many of our partners, including Lincoln Laboratory, which is part of MIT, we have very unique system level and prototyping level expertise. We have access to state-of-the-art facilities all the way from MIT Nano for microelectronics fabrication to CCTR for human studies, etc., that we are ready to combine and connect. And finally, we are right in the middle of the most dynamic, exciting, entrepreneurial biotech ecosystem in the world. So we have an obligation to drive this new revolution. And that's why we started what we call the MIT Program for Health Sciences and Semiconductors, or HSS, with the mission to drive innovation at the convergence of health sciences, AI, and semiconductor technology, leveraging minimally invasive physiological measurements and therapeutics to improve human healthcare and wellness. And we are doing this, as I mentioned before, by focusing on the detection and analysis of physiological signals in a way that is minimally invasive. And why minimally invasive is so important? It is important in our view because it is the only way to get the economies of scale that have driven microelectronics industry for the last 75 years. So if we can develop sensors, medical devices that millions and billions of people can use, we can then drive down the cost of those systems from today's millions of dollars down to a few dollars. We've done that in microelectronics, and we can do it in healthcare space. So this is a diagram of how we are doing it. We believe that it's important to have a closed loop approach where we combine monitoring and sensing with all the analytics and security that AI brings to enable new ways of intervention. And to make this happen, we need to work at a full stack approach. We need to work closely with local hospitals and industry, clinicians, engineers, scientists. They are all part of the same team to really enable this data-driven prevention, diagnosis, and treatment that we want to enable. So we started a little bit more than a year ago, and we've been quite busy ramping up different activities, initiatives, programs, etc. Here I have a summary of some of them. We've had at least 3 different symposia and internal meetings. We've started several classes aimed at highlighting the convergence of the 3 fields that we've been talking about. We are also putting together a state-of-the-art laboratory to prototype these new medical devices, what we call the waves, beats, and molecules lab. Waves because of sensing, beats because of AI, and the molecules because of the new ways of intervention that this is going to enable. And of course, we've been working together in small and large research projects. Areas where we are planning to grow over the next few months is we want to put together a commercialization studio to nurture entrepreneurship and new startups, as well as fellowships and internships, where we really need help, and we believe it is part of the future. We had a few large-scale workshops over the last year or so. We have put together a request for proposals that has been very successful. The focus has been on basic and applied health science research that leverages semiconductor technologies with a clear path towards clinical translation. We have awarded three grants so far, of around $150,000 per grant for MIT. And this particular call was done in collaboration with our partners at IMEC. IMEC is the largest research lab in the world on everything related to microelectronics. They were founded in Belgium, but because of this HSS program, they have actually established an office here in Cambridge, and we are working very closely with them in this first call for projects. And we already have three projects that have been awarded and we are working on. The first one on new ways to quantify hemoglobin by using photoacoustics, and this is led by Professor Brian Anthony. Second one on developing new functionalization techniques for the next generation of nanopore-based biosensors. And more recently, and that one with Professor Jeremiah Johnson, and more recently with Professor Timothy Swager on new fluorescent monitoring of inflammatory biomarkers. And we expect the next call for projects to be announced in the next 6 months or so. We are also working on large research programs. Here we have one that was just funded by ARPA-H. We have a couple more under DARPA sponsorship that are starting as we speak. And now, in the last 20 seconds, the key message is we want to work with all of you. This is a once-in-a-generation opportunity to really revolutionize the future of healthcare. Thank you.
Hi, everybody. How are you today? I'm Alex Shalik. I'm the director of the Institute for Medical Engineering and Science, a faculty member in chemistry and a couple other places. And on behalf of myself and Emery Brown, who will be here for the Q&A, I wanted to introduce another program that builds off many of the things that you've been hearing about so far in Tomas's speech, but also in Joe and Elazar's, where we're really trying to figure out how to do better partnerships and closer partnerships with the hospitals so that we can really drive clinical impact through partnership. So I'm a big believer that MIT is incredible. I don't have to tell you about all the facilities, all the students, all the colleagues, but what makes MIT, bar none, the best institution to do the kind of work that I do in my mind is this ecosystem that Tomas just referred to. It's the fact that we're placed within this incredible community of other universities and institutes, healthcare systems, incubators, and biotech. And what's really truly unique is sort of the density of hospitals and medical systems that surround us. And so, as somebody that has evolved to engineering, not realizing that I was not a scientist, I'm actually probably an engineer, one of the biggest things that I realized is that we have to identify the right problems to engineer solutions towards. And really, the goal of this program is to leverage these key stakeholders in partnership to identify the right problems so that we can drive impact. So the overarching goal of the MIT MGB seed program is to foster deeper collaboration between MIT scientists and engineers and MGB clinicians and researchers to really accelerate health innovations, very much in line with what you heard thus far. And I'll say that this is particularly important to me as a faculty member both at MIT and MGB, and to Emery as a faculty member at MGB and MIT. And our goal is to develop cutting-edge tools. This can be diagnostics, therapeutics, digital tools based upon AI to really address critical challenges that come up in the hospitals. Now, my favorite part about running a seed program is that I get to have money where I can go out and I can support my colleagues in tackling really important clinical challenges that exist at the boundaries between these institutions, centering real-world applications that can impact patient care. And we get to integrate together all the amazing things that happen across the various departments and programs here, from AI to ML to sensing, to really improve the delivery of care. And we get to think about all the other things that MIT has done really well to build out the entrepreneurial resources that will really enable us to not just do research, but drive impact. Now, the great part is that we, thanks to a gift from ADI, have funding that we can use to support a number of faculty members. So our seed grants, we started off with $2 million per year. The goal is to have at least one faculty member on both sides, and a focus is primarily on AI and healthcare, diagnostics and novel therapeutics. We tried to keep the proposal short. We tried to make it balanced in terms of funding. And with the generous support of MGB and MIT, we raised even more money, so this is fantastic. But the big problem for Emery and me is that when we went to actually put up the first set of proposals for this, we received 134 of them. So it quickly went from this idea that we were going to be well loved by our colleagues for everything we could do to, oh my God, we're not going to be able to fund everybody that we want to fund. So this has been an incredible program. We got this large number of proposals. We worked through the initial review as a team. We did short pitches with Q&A. And then we selected the top set of proposals, and honestly, I would have funded so many of them, but unfortunately, we only had so much money. So the things that were just recently awarded and that are now kicking off sort of break out into 3 buckets. The first one of them is really around devices, and these range from things that are incredible, like the construction of a bionic nose, to ways of improving MRI so that portable systems can give you the resolution you only have in hospitals, to devices that enable you to think about what is the right drug for a patient by giving microdoses, to ways of predicting risk for sickling and sickle cell disease, to prosthetics that can actually feel. So you could imagine in reading through this stack, it was really, really hard to figure out what should be included or not included. On the AI and computing side, we had an equal number of amazing proposals, from using ML on EMRs as a way of figuring out early endometriosis prediction to make sure that we had impact as early as we could on individuals, ways of improving design of binders for anti-tumor therapies, ways of predicting neurocognitive decline through speech analysis, and ways of improving things that happen in histopathology, of using ultrasound to improve placement of devices and precision medicines, and finally, trying to even do things in the supply chain where we can think about how to improve the likelihood that an organ from a donor actually made it to a target population. There were equally amazing things in the therapeutics and diagnostic space, from new AI strategies for developing antibiotics towards things like Pseudomonas, ways of targeting damaged extracellular matrix through manipulating collagen, new classes of cell-based therapeutics and diagnostics for inflammation, ways of figuring out interesting connections that people have seen between GI pain and autism spectrum disorder, and then finally, patient-specific avatars as a way of predicting who was going to respond and in what context. So I just want to take a minute to say that this is the first cohort of awardees. I couldn't read through everybody's names, but we're extremely excited and impressed by this broad group of people that represent not only MIT and MGB, but many of the surrounding teaching hospitals, and Harvard and Broad and the Reagan. And we're really excited to see what will happen, not just when these people kick off their projects, but when they start to interact with one another and when their students start to interact with one another and bring us the best ideas. Because in all honesty, the people that have the best ideas here are the students. We're just helping to increase the number of random collisions. So we do have plans now that we have this first set in place. We're going to launch a next set of proposals. I hope that we get just as many and hopefully not many more proposals, but I hope they're all equally as good. We're going to have an annual symposium that we're planning right now, as well as sort of monthly research meetings that go back and forth between the institutes as a way of creating a tighter bridge between MIT and MGB. We really want to make sure that what we do doesn't become problems that were discovered and brought to MIT to die on a shelf. So we're really thinking about who do we need to have at the table through an advisory board and through the surrounding ecosystem to make sure that anything that we're doing actually delivers impact. And most importantly, we had 134 proposals. I won't lie to you and tell you I'd fund every single one of them, but I would have funded easily over 2/3 of them because they were all amazing. So we're really looking to raise additional support so that we can bring more people into this program. So I'd say stay tuned. We'll be launching a series of internal meetings. If you're interested, please let me know. We'd be delighted to get you on the list. We'll be having a larger symposium, very much like the ones that were just announced for the Health Sciences and Semiconductor program. And we will have another call for proposals. So we're gearing up to get all of that started. And I'll just take a moment to say thank you to everybody. This was a very complicated program to pull off, and we only played a small part in it. We're really appreciative to the Office of Innovation and Strategy, the Office of the Provost, the Office of the President, OSAT, School of Engineering, School of Science, Resource Development, MGB, and Analog Devices that made something like this possible. And the thing that keeps me going as I think through this is seeing the incredible response to a call for proposals here, telling you how much our ecosystem really wants this. And so we're delighted to continue pushing this forward and to see all the amazing things that will come of it, and look forward to inviting you to see the products of that at our symposiums. And so with that, I'll turn it over to the next person. Thank you.
So now for something a little different. I'm going to tell you about menstruation science, which is less of a program that you apply to get funding for a single project, and more an international movement that MIT is leading to completely destigmatize menstruation by making it into a science we can talk about anywhere without stigma. The same way that breast cancer went from something you didn't talk about at all to something that is very well funded and that nobody associates with tit or boob anymore. Our dean can talk about it and our provost can talk about it because there were very active patients together with publication of this seminal book that changed the equation and brought fantastic scientists and clinicians together to change the lives of patients. So what I'm going to talk to you about actually preceded HEALS, and it was funded by a generous gift from Emily and Malcolm Fairburn. But now it encompasses much of the MIT campus, industry partners around the world, academic partners around the world. So I'm going to tell you a little bit about how it came to be and where we're going and how it fits into HEALS. So I want to start because all over MIT you will find people driven by trying to slay a dragon that almost got them, and that's certainly true in my own case. Many of you in the room will attest that the maternal instinct is a force of nature, and it will cause you to do the most amazing things, lift a car off a child that might get hurt, etc. In my case, I failed IVF, and I failed IVF because I had a disease that was incredibly painful and debilitating starting when I was 12, called endometriosis. Endometriosis is characterized by the growth of tissue that resembles the endometrium outside the uterus. It shows up in lesions. It can be on the diaphragm. It invades the bowel. It can cause terrible pain, terrible GI symptoms, infertility, and many other things. And what we know is it afflicts a lot of women, but we must be inclusive. It also afflicts girls, and it's a pediatric disease and that's not well recognized. I wouldn't be talking to you here if therapies for this were readily available and effective, but they're not. And I got pulled into this partly because my own niece, who grew up 30 years after I did, was diagnosed as trying to get out of going to school when she actually had stage 3 endometriosis diagnosed finally at age 16. Lava shot out of my head, and I answered the call from the surgeon who had operated on me, Dr. Keith Isaacson, who was then at Newton Wellesley. We got a foundation grant, but he told me a huge problem is the drugs we have haven't changed in decades. Animal models do not work. We need engineers to come in and figure out a better way to develop drugs. Well, that was my cup of tea because I'm known for inventing ways to grow human tissues on microfluidic platforms. However, so we founded together with a gift from an anonymous foundation, this center to bring together our community at MIT to bring new approaches into this disease. And a very critical part of that was not just my experimental methods, but the systems biology from Doug Laufenberger, the founding department head of biological engineering. Now, I'm going to buzz through a few things we did and then tell you where we're going, because it's really exciting. We went in not knowing anything. We did a study of peritoneal, the abdominal fluid, and we identified, using machine learning 15 years ago, the first molecular classification and a new potential target that wasn't hormones, something called Jun kinase. Very excited. We found out that a scientist, Steve Palmer, at a local drug company, had already been getting them to do preclinical models of a drug they were developing for something else. Those worked, but when they went into clinical trials, the trial was not successful. I met up with Dr. Palmer at a conference in Hawaii. There was a hurricane, so we spent some time thinking about the future of Jun kinase inhibitors. And he told me he would go invent new drugs if I would invent better ways to test them in patients. And it had to be something that was like a lesion. You see that group of cells forming a little cyst, immune cells, and blood vessels. And that's what he wanted, and he wanted it to be from patients. Okay, so we had a lot of work to do. We got a huge DARPA grant and a lot of other industry grants to piece together the things we needed. And one thing is we needed a whole new way to grow human organoids. Now, we got funding to do gut organoids, but what you can see here, so on the left is the standard way biologists do it, and they grow them in something that's derived from a tumor. So this isn't very good. It's not reproducible. We invented a completely synthetic environment that's reproducible and that's now being commercialized. We had to translate that into the endometrium, and you can see here patient-derived glands, and then growing them as organoids, green, together with the red cells, which are the supporting cells. Okay, now we can go a long way to test drugs with cultures like that, but of course, vasculature, blood vessels, are part and parcel of what makes the lesion so bad. And this is a lesion in the uterus of a patient in her muscle. So how do we grow the vasculature? Where our colleague Roger Kam had invented a way to inject blood vessel forming cells inside of a gel into the center of a microfluidic device. This isn't it's hard to see, but what you see are the green cells or immune cells, and they're moving from one channel to the other side of a microfluidic device that's been commercialized by the Kam Lab. The device is shown there. What we did, working with Dr. Kam on an NIH grant, is we adapted that approach to add the endometriosis lesion cells. So what you see in this movie here, and it might be a little bit faint given the lighting here, is a time lapse over 6 days. We injected the cells in together, so it's starting here, and what you'll see is the cyst growing bigger, but blood vessels are forming in real time to nourish it. Okay, and so this paper is in revision. We anticipate it will be accepted any day now. And this sets the stage now to go back and test drugs for efficacy in a model of the lesions themselves, because what you don't see are also immune cells in there. Now, that's all great. It's in my lab, and my lab is a really fun, happening place with talented people. But what we do in lab isn't really that impactful unless we translate it out into the labs of other people, both academic and industry investigators. So we are working very closely with Dave Trumper's lab and some local companies to translate these prototypes that we have in our lab, because we must include continuous pumping and ways that can circulate immune cells and drugs. And we have prototypes that will be ready to translate to our collaborators late this year and probably into commercialization sometime next year. Now, endometriosis exists in your body, which is connected to many other organs and affects other organs. And when you give drugs, those drugs also affect the other organs, not just the target tissue. We can apply these same approaches to build little microvascularized models of liver. We've been working with Novo Nordisk about 8 years on these kinds of problems for metabolic disease. And here is from a paper we just recently published showing vascularized liver where immune cells can circulate, and it's a model of insulin resistance. But this is also very important for drug development for endometriosis, because no matter what drug, if you have a Jun kinase inhibitor, you ultimately have to test it in liver, hopefully a female version of liver, to see if it's safe to give to patients. And I have here a paper, actually a very nice paper from colleagues at Bayer who had an endometriosis drug fail. in clinical trials, and it was very sad because it was liver toxicity. Now, this field of growing things on microfluidic platforms is called novel alternative methods, because we want to use human models, not animals. And our new NIH director was at MIT last fall talking about his real support for this, NAMS, and he has invested a lot of money in it, created a new office, and there's a blueprint for how NIH is doing this. And Kelly Pate, who heads our animal care facility, she's a vet, and I co-wrote that report for NIH. And I can guarantee you endometriosis and chronic Lyme are in that report. Now, because of the support we had from the Fairbairn Fund and others, philanthropic support that let us do things that we couldn't get funding for otherwise, we became competitive for a whole new NIH NAM center that's going to focus on applying these kind of technologies, particularly to endometriosis, but sex-based NAMs, interacting with clinicians and geneticists to take clinical data, design the in vitro models, and benchmark against what the patient samples really, really are. So this is a huge program, and it involves a lot of people at MIT and at other institutions and around the world. Our flagship project will be to test new Jun kinase inhibitors that Steve Palmer, shown here on the left, developed and that are now licensed to Gideon Richter. And we are very grateful to Emily Fairbairn, who we call a citizen scientist. She had chronic Lyme. She also had endometriosis, and she went in to fund research that she thought would move things forward faster than you would ever get from NIH. And we interact with her almost daily around ideas she has, and it's been a terrific, terrific interaction. So I'll close by saying our goal is to bring great new science and technology from MIT to study basic menstruation processes, because menstruation sets immunology for women across many diseases, not just endometriosis, but everything else. And so it's a lot of fun. We have many events, and we'll be having many more, and you can sign up for our newsletter. And I will thank all my collaborators who have made it possible for us to launch this and to engage the community and other people, and that actually is the end. I'll go back.
We invite our panelists to come on up. Have a seat. Really exciting things that we just heard today. The presentations you've seen, they all share a common thread. Each one is grappling with what it actually takes to move promising ideas out of the lab and into clinical practice, from pediatric innovation to semiconductor-enabled medicine, from seed partnerships with Mass General Brigham to closing long-standing gaps in women's health. These efforts represent different answers to essentially the same question: how do we make the journey from bench to bedside faster, fairer, and more durable? So I'd like to use the rest of our time to pull back the curtain on that. And so I'm going to ask the panelists, I have two questions, if we have time, a third, and we'll just go down the row and then back again with the second question. So the first question is, has direct contact with a patient or a patient community ever fundamentally changed the direction of your research? And what does that look like in practice? So we'll start with Dr. Emery Brown.
Well, mine certainly has, because practicing as an anesthesiologist, that's where most of the ideas come from. Actually, realizing you see how poorly or how primitive our techniques are, and then realizing that there has to be ways to improve it. Just to put that in the context as far as anesthesiology, ether was discovered as an anesthetic back in 1846. We still use ethers, basically. Cialflurane, desflurane, isoflurane, they're all ethers. But there's a lot more which is known about the brain, and we don't take advantage of that. Anesthesiologists are some of the best some of the best pulmonary physiologists, some of the best cardiac physiologists, but we don't have the same reputation in neuroscience. And so there's a huge array of possibilities there.
Well, thank you, Emery. Tomas.
Thank you. I mean, I am not a medical doctor. I am an engineer. And because of that, my interaction with patients has always been indirect. However, what I have observed as an engineer myself is that my complete approach to the question of healthcare and the clinic has changed every single time I've spent time with doctors at the hospital, learning how they do things. It's completely eye-opening, and every engineer who wants to get in this field should spend time at the hospital. I still remember the first time I joined an electrophysiology study. It was amazing, and to see the technology and how different it is from what we use in normal life. It was eye-opening. The first weekend I spent in an ICU, again, to see how doctors and nurses use all this really incredible technology that is so different from what we have outside the hospital. It's eye-opening and at the same time a huge call to action, because I know we can do better. I know that my colleagues in the non-medical space have lots of creativity and ideas, but there is a huge mismatch right now in the language and the understanding. So we need to bridge that gap, and we need to find more ways, or better ways, or more efficient ways to bring engineers into the hospital. And I think many of these programs are aimed at that.
Thank you, Tomas. Linda.
So I certainly mentioned my own patient experience and that of my family members in my talk. And the OR picture I showed you, I took myself while collecting samples from patients. And I have many patients write me every single week because I'm frequently in the news. And I live in a special place in the whole ecosystem because clinicians really respect me for the work we've done and published and changed clinical practice, and the way that we're interacting with industry to really move to make it possible to even evaluate drugs that companies wouldn't think about trying to take into the clinic a few years ago. So that's part of it. But what the Menstruation Science Initiative is to build on that and bring in the patient voice of patients who are not heard. I was just at the HHS Women's Health Conference last week representing MIT on a panel, and the first panel was actually about invisible diseases, and Dr. Mickey Tall, an immunologist who's a principal scientist at MIT and the co-director of the Menstruation Science Initiative with me, studies chronic Lyme disease. In fact, she runs MIT's largest clinical study. It's to study chronic Lyme and long COVID. And these kinds of patients have not been heard, and their symptoms are difficult to measure. They are difficult to sort through and find themes because there's all different groups of patients. And so one of the things that MIT can do is engage directly, and is doing, and what our initiative is doing, is engaging directly with what we call citizen scientists, people who have some pretty good ideas about what's really wrong with them, and listen, find the themes, find ways to measure things we can't now measure about brain fog and so on, and put these together to come up with ways we can do mechanistic studies with the tools I talked about. We're very optimistic. We're going to have a lot of events coming up that will engage with patients, because patients really know a lot when you give them the opportunity to correspond with you in a structured way.
Thanks, Linda. Joe.
Well, thank you for that question. I like the question because it gets to the heart of why I'm here. My answer is that every meaningful innovation that I can think of was born sometime in the middle of the night as someone sat next to the bedside of a patient and asked, why can't we do more? Why can't we accomplish what our goal for this patient is? And why is there not a technology or some process that could make them better? I think that if I think back, over the years, most of these innovations came from that kind of ethos. Someone sitting at the bedside and wondering, what more could we do? For instance, when I was a fellow sitting at the bedside with a patient who had a little baby who had persistent pulmonary hypertension, who was dying from their lack of circulation to their lungs. And we had no way to directly affect that. But during our fellowship, we pioneered the use of nitric oxide for these patients to increase the blood flow to their lungs with a simple gas. Other innovations that were dreamed up at the bedside, like the Otis valve that my colleague Elazar Edelman helped to make with colleagues at Boston Children's, that came from the thinking that a child born with a valve defect needs 3 or 4 surgeries as they grow. Isn't there a way that we could build a valve that grows with that patient? This was the nidus of how it worked, of how it started. And the reason that bedside clinical insight is so important in this innovation, especially in pediatrics, but I think across the board, knowing what's happening with patients, asking the questions at the right time, and getting people really enthused about the possibilities in the future.
Thank you, Joe. So we have a number of ears in the audience. We have academic leaders, policymakers, people from companies and VCs. So a second question is, if you could point to one thing, a policy change, a new kind of funding mechanism, a shift in how academia trains our students for the next generation that would most accelerate the path from lab to clinic, what would it be? And we'll start with Joe and then move toward me.
Thank you. So another great question. I think for at least in the pediatric innovation space, a real opportunity exists for shortening the regulatory pathway to the bedside and allowing the use of real-world data to inform our innovations. So that pathway is beginning to be opened to allow us to use data that we collect routinely to analyze our innovations and to chart our path to the bedside. So I think from my perspective, the regulatory inhibitions for innovations in our field are one of the biggest barriers.
Thank you, Joe.
So I would echo regulatory barriers, and we're trying to do something about that. But I would say in my main field of gynecology, that we need to think about flipping a paradigm where we spend enormous amounts of money on fertility treatments for adults. And we don't hardly study girls as they're making the transition through puberty when they start to develop chronic problems that plague them and prevent their fertility. We could save an enormous amount of money and time and agony and tears if we were to study girls and flip that paradigm and invest more. And these are the kinds of things I think we need to speak up about, and we can't wait for NIH to do it by themselves. Industry has their hands strung a bit because they're not going to do clinical trials on girls under 18. So we as the academic community are struggling, but succeeding in finding creative ways to fund studies and fund outreach efforts to get a focus on girls so we can nip things in the bud and we can invert the effort that we're spending to treating things way down the line when it's painful and agonizing to finding them early, treating them early, and preventing those things. That would be the big thing, and I've spoken up at HHS.
Excellent. Okay, Thomas.
Again, I'm approaching the problem from the microelectronics industry and semiconductors and how do we leverage all the innovation there. And I feel there is a huge opportunity if we are able to leverage those platforms that the microelectronics industry has. If the next time we try to develop a sensor to measure some specific biomarker, instead of starting from scratch, as the great majority of programs do, we could actually start from a well-tested microelectronic platform that is commercially available, but where we may need some additional know-how on how to access it, etc. At the end, we are not doing that because of IP concerns. The companies that have that IP, that platform, they are very concerned that we may block them in one way or another. So I feel we need to work on the IP issues. From my point of view, universities typically need IP to be able to start companies and new enterprises in the future. Maybe there are other ways to do that. So IP, we need to look at the IP issue.
Thank you, Thomas. Emery.
So I'll go back to just what we were talking about in the first question. I think that we need to have more mechanisms through which our students learn about what the real problems are in the hospital. And we have clever solutions that work in the laboratory, but they're not the solution to the problems that actually exist in the hospital. And I think that, you know, one of the things, like when the HST program, is the students actually spend time on the ward seeing what's going on. And many of them say that that's the most enlightening part of the education process that they were in. And I can tell you from practicing, the sorts of questions that you think about when you're watching someone, you know, suffering in front of you differs. You know, that shapes your thinking dramatically, let me say it that way. And it provides this incredible incentive, like, to solve the problem. And I think that if we could foster more of that sort of contact with the actual clinicians, taking care of patients, and so that's one thing, but also there are tons of seminars that are held in the hospital, going from grand rounds up to research seminars, which we should feel free to go and participate in and learn, because that's where the actual problems are being discussed. Or, for example, M&M, where they discuss what went wrong. I mean, and then it's a great way to be enlightened. I think we should take advantage of those and have more contact with our clinical colleagues.
Excellent. So we're hearing everything from regulatory and policy changes to getting students back in the classroom and engaged, well, out of the classroom and engaged with clinicians, to more fundamentally looking earlier at the source of the diseases and seeing if we can prevent. I hear a lot of great ideas. We're running short on time, so I don't know if we can answer the third question. What I might do is just pose the question, and that might be a topic for conversation with the audience, which is, what do we wish people understood better about what it actually takes to move something from a promising idea to a clinical reality? I'd like to thank our speakers. We heard about some incredible programs today, and we've learned a lot about the hard work of taking something from the lab into practice in a way that really impacts people's lives. So thank you so much.
Thank you.